try to understand translational motors, microtubules play a very important role. So microtubule kinesin dynein system is another classic example of a translational motor. So in that, the microtubules forms the track on which the motor proteins like dynein, kinesin, etc. will be traveling. Now, what exactly is the structure of a microtubule? So, microtubules are assembled, it's a homodimer, it is a 50 kD, which is made up of a heterodimeric unit of alpha and beta subunit. So, these alpha beta subunits are repeated again and again. And these alpha beta subunits forms a kind of it forms something called a protofilament. Now, these protofilaments, they will be looking like this, the protofilaments. So you can, and in these protofilaments, what is important is, if this is alpha, this is beta, alpha, beta, alpha and beta. Now, these protofilaments are arranged into a cylindrical structure. Okay, it is arranged into a cylindrical structure like this. So, cylindrical structure. So, similarly, you will find this kind of another protofilament like this. There will be 13 protofilaments. If you consider this is 1, 2, this is 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 and 13 protofilaments are arranged, and that forms a kind of a cylindrical structure. Now, what is important is this alpha and the beta both have a bound GTP molecule. Both have a bound GTP. But if the GTP which is present in alpha is somewhere in this embedded, or it is trapped inside, which means this GTP cannot be hydrolyzed. But the GTP which is on the beta subunit, that is hydrolyzed, and if the beta subunit has a GTPase activity. Now you would have already seen that this portion with the beta subunit which is at the other end, that is called the plus end of the microtubule. So remember, the plus end will have the GTPase activity, and this end which begins from an alpha subunit, that is called the minus end of the microtubule. If you look at the diameter, it varies from 25 to 30 nanometer is the diameter. If the inner portion is a hollow, it is a hollow that is somewhere around 17 nanometer is the hollow unit of this microtubule. You will find plenty of microtubules inside the cell which forms a kind of a track for either protein, dynein and kinesin molecule to travel on this. When we speak about the microtubule, one important aspect is something called the microtubule assembly. The microtubule assembly. How exactly are the microtubules assembled to the form that we see? Now remember that in the microtubule, it normally begins from the centromere. So you know that in the centromere, which is closely linked with the outer envelope of the nucleus. Now centrioles are L-shaped structures. From there, the microtubules begins. If the minus end will be there and the plus end will start growing. So therefore you will see that the plus end, like a star kind of a structure, it grows, which is known as the aster. So it grows. This will be the plus end. So if the minus end is in the center and the plus end is moving towards the periphery of the cell. Okay, that is how the microtubules normally grow. In the microtubule, assembly is also called the Microtubule Dynamics. So, what exactly is meant by the Microtubule Dynamics? Look at the average lifespan of a microtubule. It varies from 1 to 10 minutes. So 1 will be where the mitosis is going on, in those cells where the mitosis is going on, if the lifespan is about 1 minute. Where no mitosis is going on, so if the mitosis is going on, if the lifespan can extend up to 10 minutes. In comparison to, remember, the alpha, beta monomeric units, they will have a lifespan of about 20 hours. So once it polymerizes, in the life it decreases in the case of microtubule. That is a knowledge that we need to acquire. How the assembly occurs? Scientists have painstakingly carried out a lot of experiments in order to understand the microtubule dynamics. What they have done is they have isolated these microtubules. It can be from neurons, it can be from any cell, it is isolated. Now after isolation, what is done is these microtubules are allowed to assemble. So let us draw a graph. Okay, this will be the time, that will be the time, and this will be the percentage the polymer formed. You will see that initially it has to the tubulins have to overcome the resistance. It has to overcome the kinetic barrier, and it will be, they are just randomly associated. And this initially, as it is getting assembled, it is what we call it nucleation. They are getting assembled, similar to the actin filaments. Once it gets assembled, then you will see that it is forming rather a long structure, and that process is called elongation. That is the second process. It is assembling into a long structure. So therefore, we can draw a graph. It is becoming elongated, and the elongation reaches something called, after some time it reaches something called a steady state. These are the three phases, that is nucleation, elongation and steady state. Steady state, if the number of units that are added is the same as the number of units that are removed from the microtubule. So this is how you get a kind of a sigmoidal curve in order to explain the microtubule assembly. So you have a nucleation initial phase, then there is a growth phase, that is the elongation phase, followed by a steady state phase. How exactly this assembly occurs? Now, in order to understand that, in the microtubules, in the alpha and the beta tubulins, they are joined one after the other with the help of GTP hydrolysis. We have seen that the GTP which is bound to the alpha subunit is not hydrolyzed, but that which is attached to the beta subunit, it is hydrolyzed and it is getting added. So if this is what you consider as a microtubule, this is the positive end and this is the negative end, what can happen is, if the microtubule, we have seen the half-life is very short, constantly undergoing turnover. So what can happen is, if the tubulins can get separated, when a tubulin is being removed, that process is called catastrophe. Catastrophe, it is a slow process and the tubulin molecules will be removed. And when catastrophe happens, you know that if this is in the microtubule, this assembly occurs, it is getting destroyed. So this is the negative end and this is the positive. What happens when there is the tubulins are attached to this? So when you attach tubulins here, tubulins are attached, you add them, then they get attached here and it will grow. And this process is what we call rescue. Rescue is growth and catastrophe is its destruction. Now this process of catastrophe and rescue will keep on happening in the case of microtubules. So if we draw a graph from here, so only this portion, if you draw a graph, you will see that over a period of time, this is a polymerization, percentage polymerization. You will see that this is growing, okay, and then the catastrophe begins. So this is the place where the catastrophe begins. Then rescuing will happen. This is the place where rescuing will happen. And again there can be a catastrophe, again rescuing, again catastrophe, rescuing, catastrophe, rescuing, like that. So you will find it is not continuously which is growing, but you will get very fluctuating graph in the case of the turnover of a microtubule molecule. One important aspect that we need to keep in mind is when it is adding tubulins to the plus end. Remember, it has got a bound GTP. If the GTP is not hydrolyzed, it forms something called a GTP cap. A GTP cap is important because it will protect the molecule. It will stabilize the microtubules, so it will keep growing with the help of this GTP cap. So this is more or less how the dynamism of microtubules happens regularly within the cell. Now, there is a protein which is associated with the microtubule, that is called the microtubule associated protein. Microtubule associated protein is, there are many forms of this microtubule associated protein. What happens to microtubule associated protein is, if you consider this is a microtubule, this protein has two domains. One domain is embedded inside the microtubule and another domain project outward. So all these studies were carried out using electron microscope to see. So they could see that they are just having one domain which is embedded within the structure, another domain which is projected outward. Now the MAP proteins have got, the MAP has several functions to perform. First of all, it will increase the stability of the microtubule. Second, it will help in arranging, parallel arrangement. Helps in the parallel arrangement of the microtubule. Third, it will promote assembly. So these are some of the functions of microtubule associated protein: increase the stability, arrange them in parallel form, and promote the assembly of the microtubule. Now, MAP is also highly regulated through phosphorylation and dephosphorylation. So if MAP is phosphorylated, that promote disassembly of the microtubule. So a dephosphorylated MAP is required to maintain the microtubules in a stable conformation.